Very good afternoon to one and all. So today we'll start with a new topic, that's the infectious diseases. So in this infectious diseases, first we'll discuss how these microorganisms causes the diseases, how this microbes enters the body and gets disseminated to various sites, and along with that how the various various microorganisms gets interact with the host and then escapes from the host immune system, as well as the mechanism of both viral and the bacterial injury very briefly, because this will be dealt in detail in your microbiology. As well as in the infectious diseases, the three most important which will be very commonly asked in the exam: tuberculosis, leprosy, syphilis and actinomycosis, we will discuss in detail. So let us first discuss few general features of these infectious diseases. So as we all know that for various infectious diseases there is an effective vaccines as well as antibiotics are available, but still it plays an important role in the causation of mortality in the both developing world as well as the developing countries. But in developing countries, additional burden to the cause of mortality due to infectious disease is the malnutrition. So especially these infectious disease are important cause of mortality in elderly people and as well as in immunocompromised individuals, or in individuals who are suffering from a chronic diseases or receiving a immunosuppressive drugs. But in developing countries, the contributing factors for these are the malnutrition, as I said, especially in the children, and in unsanitary living conditions also play a role in the causation of mortality due to infectious diseases. But today there, because of this extensive research in the molecular genetics as well as of the entire genomic sequence of many species including microbes as well as humans are known, this may promise for a future research in the pathogenesis, diagnosis as well as in the treatment of various infectious diseases. So coming to the certain terms which needs to be understood are that, so what is infection? Infection is nothing but the infectious agent has entered the body, but it start replicating, but it may or may not cause a disease. So infection means that the pathogen, it is present within the host and it starts replicating, but it may or it may not cause a disease. Whereas pathogen is a infectious agent which will cause the disease. Okay, and whereas disease is a term where there will be a clinical features of signs and symptoms of that particular disease. So all infections may not cause a harm to the host. Okay, and Louis Pasteur and then the Robert Koch, both of them are a pioneer. Robert Koch as well as the Louis Pasteur, who they are the pioneer in establishing the various microbiological etiology in the infectious diseases. So as you all know that Pasteur is the one who created the first attenuated vaccine against the rabies, whereas the Koch is a criteria for linking specific microorganism to a particular disease. And as we know that there is a Koch postulate which says that the organism must be found in the lesions of the disease, and that organism should be isolated as a single colonies on solid medias, and if these organisms is inoculated on experimental animals, else that should cause the lesion, and the organism can be again should be recovered from the experimental animal. I'll repeat, this Louis Pasteur and Robert Koch were the pioneers in establishing the specific microorganism or a microbiological etiology to the particular infectious diseases. And Koch postulated that the organism must be found in the lesions of the disease, and the organism must be isolated from that particular lesion and as a isolated colonies or a single colonies on a solid media, and those colonies, when it is inoculated on an experimental animal, it should cause a lesions, and from those lesions the organisms must be recovered from an experimental animal. Next, coming to the types of the microorganisms, the infectious agents, they belong to the wide range of classes. It may vary in size from kilodaltons to nanometers to meters. So which composed of a prions, virus, bacteria, fungi, and then the parasites. Then how these microorganisms causes a disease. So as we all know that, coming to the modes of the entry of the microbes can enter the host by the various routes. One is by the skin, another GIT, respiratory tract, urogenital tract, and then the vertical transmission. And through the skin by inoculation, what that occurs either by the breaching of the epidermis or by the mucosal epithelium, by the fatty acids, antimicrobial fatty acids, and the deficits that are small which are toxic to the bacteria, which may initiates the mechanical injury of the epidermis. And next is by the GIT, that is by the ingestion, mainly by the fecal matter, which is contaminates the food and water and results in especially the diarrheal diseases. And further, the respiratory tract by the inhalation. Most of the organisms are cleared by the upper respiratory tract by the mucociliary actions. Some of them, that is the less than 5 microns, which reaches the alveoli, and where their role set is taken up by the alveolar macrophages and then the neutrophils, which recruits in the lung, and it is cleared by the alveolar macrophages. And in the urogenital tract, as we all know that the urine, which is very sterile, and regular emptying during the micturition clears the pathogen. So the disease occurs whenever there is an obstruction to the urinary flow or reflux of the urine compromises the normal defenses that may increase susceptibility to the urinary tract infections. Or next is the sexually transmitted diseases. Though there is a protective flora in the vagina and in the cervix, rectum, or pharynx, so this usually the organisms cause these infections tend to be short-lived outside the host. So they usually depends upon direct person to person spread. Then coming to the vertical transmission, it may occur either in the mother to fetus or the newborn child, which it may occur during through the placenta, or it may occur during the birth, where the birth canal will be, where the infectious agents may be present within the birth canal, or in the postnatal transmission that occurs through the maternal milk, like cytomegalovirus, HIV, and then the hepatitis B virus. Then this is just a the picture depicting the various modes of transmission of a microbes to a host, either by the body contact, sexual contact, to the food and water, as I said, contaminated food and water with fecal material, droplet or airborne, that's the respiratory tract, or by carrying a vector, even that can cause a disease, that's the malaria, and injecting the vector. Next, coming to this, once it enters into the body, how these organisms gets disseminated in the body. It can disseminated either by the hematogenous route or by the lymphatic nerves or by the transcelomic spread. But among these, the most common mode of disseminating throughout the body is the hematogenous route. But in certain conditions, even these microorganisms can travel or disseminates along the nerves, that is the peripheral nerves, and then through the intracellular axons it may causes it may traverse into the CNS, especially the viral infections. And then coming to the transcelomic spread, most of the certain infections may disseminates throughout the body through the transcelomic spread. But among these, the most common routes of disseminating of these microbes in the body is the hematogenous as well as for the lymphatic spread. Then further, how these microbes, once it gains entry into the host, interact with the host or gets escaped from the host immune system is that one is by the host defense against the infection, another one is by the immune evasion by the microbes, and last but not the least is the host immune response effects. So first is the host defense against infection is that as in a human both innate and the adaptive immune system are there which tries to prevent or eradicate the infection. So whenever there is an diminution of the host immunity, that results in the aggressive reactivation as well as the disease. Then to the immune evasion by the microbes, there are by various forms the microbial agent or the microorganisms tries to get escape from the host immune system. First and foremost is by the antigenic variation. So as we all know that whenever the pathogen enters the body, it has to enter through the certain specific cell through getting attached to the specific cell surface receptors. So to prevent that, so once it enters this thing, it tries to change its antigenic presentation on its surface, thereby it gets escape from the antibody mediated host defense mechanism. Or next is resistant to antimicrobial peptides by inhibiting the peptide binding or by producing the proteases to degrade the peptides. So next is the resistance to killing by the phagocytes, like it may either kill the phagocytes itself or prevent their migration towards the phagocytes, or it may diminish their oxidative burst. So three things what it does is it gets resistance to killing by the phagocytes. One is by either by preventing the migration or by diminishing their oxidative burst, or it may be resistance even to the intracellular killing by the phagocytes. It remain within the phagocytes and keep multiplying for a longer duration. Then evasion of the apoptosis and manipulation of the host cell metabolism. So so whenever it gets escape from the apoptosis or from the host cell metabolism, it allows continuous replication, and this enters into a latent infection, and even sometimes it may even transform the cell into a neoplastic. Or sometimes it may resist the cytokine or the chemokine or a complement mediated host defense mechanism by interfering with the actions of these cytokines, chemokines or the complements. Then evasion of this recognition by the CD4 helper T cells and the CD8 toxic cells, either by binding to or by altering the localization of the MHC complex thereby impairing the peptide presentation either to CD8 or the CD4 cells. So I'll repeat. So the immune evasion or the escape from the host immune system by the microbes occurs by the various ways. First and foremost is the antigenic variations. So by altering the expression of the antigens on its surface, it escapes from the antibody mediated host defense mechanism. And resistance to the antimicrobial peptide killing, that is by inhibiting the peptide binding or by producing a proteases, that is enzymes, to break down these peptides. Next is by the resistance to killing by the phagocytes by preventing the migration or by diminishing this oxidative burst or by resisting the degradation within the cells, and it keeps multiplying within the cells. And evasion of the apoptosis and then the manipulation of the host cell metabolism, thereby it allows the replication as well as it enters the latent infection. And resistance to cytokine, chemokine and a complement mediated host defense by interfering with their actions. Then last but not least, by evasion of recognition by the CD4 and then the CD8 cells by altering the MHC class of proteins, thereby impairing the peptide presentation to these CD4 and then the CD8 cells. Or last but not the least is that it may establish a state of a latent infection in which few of the, if any, viral genes are expressed. That's called as when the immune system is to lie low. Next, what are the morphological spectrum of inflammation or a host response to this particular following the host microbial interaction it occurs is that usually this host the pathogen causes the disease either by the causing a cell death or by causing a cell injury or by releasing the toxins and thereby causing a destruction, or it may causes the so this pathogen either the when once it enters the host, it may cause a disease either by causing the directly the cell death, or it may causes the releases the cellular toxins, or it may causes the various enzymes which may causes the destruction of the tissues. And then okay coming to the various morphological spectrum of the infection. One is by the suppurative, it may cause suppurative inflammation, which causes the extensive acute inflammatory response, which leads to the accumulation of the neutrophils at that particular site, and then followed by mononuclear and then the granulomatous inflammation that occurs usually especially in response to virus or to the intracellular bacteria or to the intracellular parasites, where the mononuclear cells will be predominant with the inflammatory lesion, depends upon the host immune response, and these mononuclear collages gets transformed into epithelioid cells, and these in turn fuse to form a multinucleated giant cells. Then cytopathic and then the cytoproliferative effects, especially seen in the viral infections, where there may be a very sparse inflammatory cells, and these virus they replicate within the cells and they cause a viral aggregates which may visible as inclusions, or it may fuse and then form a multinucleated cells called as a polykaryons. And tissue necrosis, it is mainly due to the secretion of this powerful strong toxins by the microorganisms, which causes a rapid and a severe necrosis and that causes the tissue damage. Later, the chronic inflammation and then the scarring. Many inflammations elicits a chronic inflammation which can lead either to complete the healing or there may be a extensive scarring. These patterns of tissue reactions, they help in analyzing what type of infectious process is going on, and they rarely appear in a pure form. Usually there will be a overlapping of these morphological spectrum in a microbial infections at a same. Then how this infectious agent causes a disease in the host. So first and foremost, as I already said, that this these pathogens, once they enter, they may cause the tissue damage or the infection or a disease in the host, either by entering the host cell, it may cause directly the cell death, or it may alter the cellular metabolism, or it may even cause the proliferation and then the transformation of that particular cell. And then next is that either by releasing the toxins, it may be a exotoxins or it may be endotoxins, or by releasing the various enzymes, it may causes the tissue destructions, and then it may causes the damage to the blood vessel, causing a ischemic necrosis, and then induces the host inflammatory cell response directly or indirectly. It may causes the injurious tissues. So these infectious agents, they cause damage either by entering the cells and directly causing the cell death, or by altering the cellular metabolism and thereby leading to a proliferation and then the transformation of that particular cell, or it may release the toxins and then causes the tissue damage, or by causing releasing enzymes which causes the tissue destructions, damage the blood vessels leading to ischemic necrosis, and then inducing the host inflammatory cell response, which will directly or indirectly causes the tissue injury. Then so few words about how these viruses and bacteria causes an injury to the cell. Viruses has got a tissue tropism that will dictate which tissue will be injured. So coming to this, how this virus is enters or and enters the host and then causes the an injury to that particular cell. So first and foremost is that if you look at this picture, the virus bind to a specific cell surface proteins. On binding to the cell surface receptors, it gets enter and gets uncoated and start replicating with the help of this messenger RNA, and then the viral protein is synthesized, and these viral proteins, it gets it may causes the or alter the metabolic derangements, or it may causes a cell lysis, or it may causes the cell fusion, or it may even start replicating and transforms that particular cell into a neoplastic cell, or it may these viral antigens may aggregate and they may come to the surface and present to a appropriate antigen present cells and then initiate a host immune response, or they may reduce the host cell DNA as well as the RNA synthesis within the cell. So, and thereby it causes either direct cytopathic effects, or it may induce the antiviral host immune response, or it may initiate the cytotoxic T cell or a NK cell host immune response. So direct cytopathic effect by inhibiting the host DNA, RNA or a protein synthesis, or by producing the degradative enzymes or a toxic proteins, induce the apoptosis, damage the plasma membrane, or it may even lyses the cells once it is inside the cell. Next, coming to the, I'll repeat. So coming to the bacterial injury, usually it depends upon the virulence of the bacteria, that by thereby it decides the ability of these bacteria to get adhere to the host or a tissue, or it may even invade the cells or the tissues and delivers the toxins. Thereby next it get adhered to the host cells with the help of adhesins that is present on the bacterial surface. Next, by producing the bacterial toxins, there is an endotoxin or a exotoxins, and endotoxins are the lipopolysaccharides which are present on the gram-negative bacteria, which may stimulate both host immune response as well as it may cause a injury to the host tissue itself. And exotoxins, they are the secreted bacterial proteins which may cause both cellular injury as well as the disease. Next, to the various techniques to diagnose these infectious agents, the most important gold standard technique is the culture, followed by the biochemical, serological and as well as the molecular. So the recently the nuclear amplification test like PCR, and then the nuclear amplification test like PCR, and then the DNA amplification test are there in the various helps in detecting the various infectious agents. So some of the techniques which may demonstrate certain infectious agents are the gram stains, most bacteria, acid first stain for the Mycobacterium and Nocardia, silver stains for fungi, Pneumocystis, PAS for fungi and amebas, mucocarbamine for a cryptococci, Giemsa for leishmania, malaria parasites, Acamplopter, antibody probes, cultures and DNA probes for almost all the classes. So this is about in general about the various infectious agents. So what we all discussed, what is infection, what is disease, and then how these microbial organisms gets access into the host, what are the various microorganisms and how these microorganisms gets entered into the host. Once it enters the host, what are the how it tries to get escape from the host immune system, and then following this how it disseminates throughout the body, and then host pathogen interaction, and that is if it gets escape from the host immune system, if it doesn't, how it causes a injury in the host, and briefly about how the viruses and the bacteria causes an injury. So now let us go into the discuss in detail the mycobacterium. So I think in the microbiology that mycobacterial tuberculosis already dealt or not, I don't know. So let us first discuss. This can be your long essay also. So this tuberculosis, it is a chronic granulomatous pulmonary as well as a systemic disease caused by the Mycobacterium tubercle bacilli. So tuberculosis, it is a chronic granulomatous pulmonary as well as a systemic disease caused by the Mycobacterium tubercle bacilli. Then what is this Mycobacterium tubercle bacilli? Characteristic is that they are slender intracellular aerobic bacilli which may grow either in branches or in a straight chains, and it is acid fast. That is because of it has got a unique waxy cell wall, that is a mycolic acid, which will retain the stains, and once it is stained, it resists the discolorization or a decolorization by the acids and then the alcohols. Therefore it is called as a acid fast, and it is weakly gram positive. So mycobacteria, they are slender aerobic bacilli which grows either in a branching or a straight chains, and it is intracellular, and it has got a very characteristic cell wall, that is a component, that is a mycolic acid, which resists the decolorization after stained by the acids and then the alcohols. Therefore it is called as a acid fast, and they are weakly gram positive organisms. Then coming to the epidemiology, and today in the world, even after various eradication programs are dealt to eradicate the tuberculosis, after HIV, tuberculosis is the leading cause of death in the world, and usually it occurs in the poverty, overcrowding, or in chronic debilitating illnesses, or in immunocompromised individuals. So along with that, as among this, the HIV is the most important risk factor. Then coming to the most important source from where this Mycobacterium tuberculosis is, where it gets transmitted, is that the open cases of the pulmonary tuberculosis or active pulmonary tuberculosis. So coming to the modes of transmission, it may be transmitted either by the inhalation or by the ingestion or by the inoculation. Coming to these three, inhalation is the most common mode of a transmission, which gets originated either from the active open case of the tuberculosis. It may be expelled either by coughing, sneezing, which creates a aerosol respiratory droplets. When individual inhales those droplets, it lodges in the lung and then causes the infection. So among these, inhalation is the most common route, which gets accessed with the active open case of the tuberculosis, which may be these tubercle bacilli may be expelled either by coughing, sneezing, which creates the aerosol droplets, which is inhaled by the individual. These droplets are inhaled by the individuals, which gets lodges in the lung and then causes the infection. Then coming to the ingestion, at present it is very rare mode of a transmission, because usually it occurs due to the consumption of the non-pasteurized milk from the infected cows contaminated with the Mycobacterium bovis. But today it's almost eradicated. So one more thing which can occur is mainly due to the swallowing of a infected sputum by the individual itself, which results in a causation of the tuberculosis of the intestine. And inoculation, extremely rare mode of transmission, but it may occur during the postmortem examination while cuts resulting from handling the tuberculosis infected organs. So the most three common modes of transmission is inhalation, ingestion, and inoculation. Among these, the most common mode is the inhalation, followed by ingestion, which is very rare, but usually it occurs due to the swallowing of its own infected sputum. Inoculation is extremely rare, which may occur during the postmortem examination, which occurs as cuts while handling the tuberculosis organ. Then coming to the pathogenesis, today we will restrict ourselves only to the primary tuberculosis. Okay, so usually this the pathogenesis in a unexposed and immunocompetent persons for the first time. That's what we call it as a primary tuberculosis. So whenever the tuberculosis occurs in a person who is immune competent and during a first time, that what we call it as a primary tuberculosis. Okay, so this depends upon the three factors. One is the amount, the number, the quantity of the Mycobacterium tuberculosis organisms which are inhaled, and then its virulence of those organisms and the host immune response to that particular organism. So it may cause either cell mediated immunity or type 4 hypersensitivity reaction. But both these mechanisms, that is the cell mediated immunity and type 4 hypersensitivity reaction, is mediated by the T helper 1 cells, which stimulate the macrophages to kill this bacteria. I'll repeat, the progression to this clinical disease in an immune competent and a previously unexposed person, that what we call it as a primary tuberculosis, it causes two immunological response that depends upon the quantity of Mycobacterium tuberculosis organisms in it, or it's the virulence of these organisms and the host immune response. It causes either cell mediated immunity or type 4 hypersensitivity reaction, so which can occur, which is mediated by the T helper 1 cells, which will activate the macrophages to kill the bacteria. So let us discuss about the pathogenesis. So as we said that it will cause cell mediated immunity, or there may be a hypersensitivity to these tubercular antigens, which will ultimately leads to the granulomas, caseation and cavitation. Then how this microbes, once it enters, gains enter into the body, it causes the granulomas. Let us discuss in detail. So in the first 3 weeks of the exposure, after the exposure, initial infection which occurs by the Mycobacterium tuberculosis in a non-sensitized, that's what we said initially in the previous slide itself, is called as a primary tuberculosis. So first 3 weeks after exposure, whatever the infection that occurs in a non-sensitized individual is called as a primary tuberculosis. Okay, so this occurs by the entry of these Mycobacterium tuberculobacilli into the macrophages, and this occurs by binding. They are mediated by the several receptors which are expressed on the macrophages. That includes the mannose binding protein or mannose binding lectin, and the CR3, that is a complement receptor 3 on these phagocytes. So the Mycobacterium tubercules, they enter the macrophages by the phagocytosis by the several receptors which is expressed on the macrophages, that is the complement receptor 3, which binds the mycobacteria, which is opsonized by the C3b. I think what are opsonins, you know, which facet the opsonins are nothing but the it is the substance which opsonizes the bacteria or the pathogen which needs to be engulfed by the phagocyte. So here the C3b receptor is opsonizes the mycobacteria, that is taken up by the phagocyte, which expresses the CR3 receptors. Another thing is by the mannose binding lectin, which is a glycolipid which is present in the cell wall of the tubercle bacilli. So next is that once replication within the macrophages. So once it is taken up by the macrophages, this inhibits the maturation of this both phagosome and inhibits the formation of the phagolysosome, which is necessary to kill the bacteria. Thereby it gets protected from these various lysosomal enzymes and not get exposed to the even to the acidic environment, and it keeps multiplying within the macrophages itself. So first it is get entered into the macrophages by binding to the receptors CR3, that is by due to that mycobacteria is opsonized by the C3b receptors, and by through the another receptor, that is the mannose binding lectin on the phagocyte. Once it gets entered within the phagocyte, it starts replicating either by inhibiting the formation of the phagosome and the lysosome, which is very much required for the destruction of the bacteria, and thereby it prevents the bacteria getting exposed to the acidic environment and results in a bacteremia and seeding of the tubercle bacilli at many sites. And this bacteria, it may be either asymptomatic or it may produce a mild flu-like illness, and it also gets protected from the microbicidal action of these lysosomal enzymes. Next, coming to the it may either initiate or enhances the innate and as well as the adaptive immune responses. So this includes the toll-like receptors, thereby this includes the tuberculosis, which composed of lipoproteins and the glycolipids. They are recognized by the innate immune receptors, including the toll-like receptors which may toll-like receptor 2 which may initiate or enhances the both innate as well as the adaptive immune responses to the Mycobacterium tuberculosis. So what are those? How does? So in less than 3 weeks of infection, which we have already said that the Mycobacterium tuberculosis bacilli, I am just depicting in a flow chart. This Mycobacterium tuberculosis bacilli either through the receptors like the mannose binding lectin, that is nothing but the lipoalbumin, or by the C3b receptors it enters the macrophages, and once it enters the macrophages it starts replicating. One is by inhibiting the phagolysosome fusion and by getting exposed to the bactericidal activity, that is acidic environment, and so during replication it causes the bacteremia which may be asymptomatic or may just produce the flu-like illness and gets disseminated and seeds into the various multiple sites. Following this, these mycobacterial antigens, they may be drained to the lymph nodes, and there it presents to the antigen presenting cells with the help of the MHC class 2 to the T cells, and here the T cells, that is to the nevi CD4 T cells, which process the antigen with the help of the interleukin 12 which is secreted by the antigen presenting cells, gets differentiated into the T helper 1 cells, and further this T helper 1 cells secretes interferon gamma and activates the macrophages to become a bactericidal. I'll repeat, after 3 weeks of infection, these mycobacterial antigens are drained into the nevi CD4 T cells, processed antigens reaches the regional lymph nodes. So here this antigen presenting cells along with the class 2 MHC which presents to the T cells, and this T cells gets differentiated into T helper 1 cells by the interleukin 12 which is secreted by the antigen presenting cells. Further, the T helper 1 cells releases interferon gamma and activates the macrophages to become a bactericidal. And further, this T helper 1 cells in the lung and lymph nodes synthesize interferon gamma and causes various transforms the macrophages into bactericidals by inducing the nitric oxide synthase which releases the nitric oxide and other reactive nitrogen intermediates and causes a bactericidal, and it activates the macrophages, stimulates the phagosome of the phagolysosome and exposes the bacteria to the acidic environment and generation of the reactive oxygen species, and thereby it becomes antibacterial, and also it stimulates the autophagy to disintegrate or the to remove all the bactericidal or the disintegrated bacterial disintegrated bacteria to remove the sequester or destroy the damaged organelles, and then the intracellular bacteria such as the Mycobacterium. Autophagy, it is a process that sequesters and destroys the damaged organelles and the intracellular bacteria such as the Mycobacterium tuberculosis, and along with that it also initiates the maturation of these phagolysosomes in the infected macrophages. So, and then further activated macrophages releases tumor necrosis factor and recruits more and more of monocytes and transforms into the epithelial cells and results in the formation of a granuloma, and then causes the tissue destruction, tissue damage resulting in a caseation necrosis. This is the hypersensitive reaction to the results in a caseation as well as the cavitation. So this is the image which is depicting that initially in the less than 3 weeks of infection, the mycobacteria which gets taken up by the macrophages through the help of this receptors, C3b receptors, and then the mannose binding lectin, and following that it inhibits the macrophages activation by preventing the fusion of the phagosome and then the lysosome, and thereby this bacilli keeps replicating within the cell itself and then leading to the bacteremia, and which results in the and disseminates to the various sites. So in this time the patient either may remain asymptomatic or may present with a flu-like illness, and after 3 weeks of infection this alveolar macrophages gets activated by presenting these antigen to the appropriate antigen presenting cell along with the class 2 MHC to the T cells, and then and then this T cells gets differentiated into T helper 1 cells by the interleukin 12 synthesized by the antigen presenting cell, that's a macrophage, and for T helper 1 cells which synthesizes interferon gamma and activates the macrophage to become a bactericidal. So this becomes bactericidal by promoting the fusion of the phagosome and the lysosome. So phagolysosome formation will be there, and then by inducing the nitric oxide synthase, thereby it releases the various nitric oxide and the reactive oxygen intermediates, and thereby causing a destruction of the bacterium, and then it generates various reactive oxygen species which is a antibacterial, and then it activates the macrophages to differentiate by synthesizing a tumor necrosis factor and differentiate into a epithelioid cells, and these epithelioid cells coalesce to form a granulomas, and along with that in the center there may be a destruction of these tissues which may be due to the hypersensitivity reaction which leading to a caseation necrosis, and later it also activates the autophagy which results in the sequestration as well as destroys the damaged organelles and then the intracellular bacteria, so resulting in the formation of the granuloma as well as the caseation necrosis. I hope I am clear with this pathogenesis of the primary tuberculosis. I hope I am clear with the pathogenesis of the primary tuberculosis, how it gates enter into the macrophages and thereby it activates the macrophages to become the bactericidal by the various mechanisms. So in the next class we'll discuss about the secondary tuberculosis pathogenesis as well as the morphology by extrapulmonary tuberculosis as well as the various diagnostic modes for to detect this Mycobacterium tubercle bacilli. Thank you.